Good evening. My senior research project is testing different types of honey's effect on Staphylococcus epidermidis. Before I start, a little bit about myself is that my name is Riley Canavan and I'm a senior at Tunstall High School. In the fall, I am attending Virginia Tech to major in biology to then pursue a career in dentistry. I chose this topic because of my interest in studying biology and medicine. Also, with my dad being a podiatrist, the majority of the patients he treats are diabetics and have chronic ulcers. I find it very fascinating but disturbing at the same time when he shows me before and after pictures of the wounds he treats. So this made me become interested in researching if honey could heal these same wounds. Also, I prefer using natural remedies for health problems if possible because of the side effects found in most antibiotics or ointments. Lastly, finding cheap but effective medicines that anyone can use is essential for combating the healthcare crisis and giving everyone an equal opportunity to improve their health. The main causes of wounds are from surgeries, injuries, and diabetic ulcers. Wounds sometimes are a tricky healing process, but when treated with honey, it has shown positive healing results. Honey significantly reduces the size of wounds when used consistently. Today, it is known that diabetes is a major health concern that has been on the rise in recent years. For example, diabetics lose feeling in their feet, which is called neuropathy, and this prevents them from noticing pain on their feet. Therefore, they develop chronic ulcers that need treatment urgently. Diabetics also have notoriously slow healing processes because of restricted blood flow. Therefore, finding effective remedies to treat these wounds in a safe way is ideal. In order to understand which honey is most effective, there are certain properties that make it inhibit bacteria more or less. The amount of sugar found in honey will determine its antibacterial properties. A study stated that honey has the ability to stop the growth of bacteria completely because of the high content of sugar. This is because high amounts of sugar will create a hypertonic environment and dehydrate cells through osmosis. Another study indicated that honey's acidity creates an environment that prevents biofilms and will release oxygen from cells, which will affect the cell reformation and result in the cells dying. Therefore, the lower the pH found in honey, the more significant healing results that will be found in wounds. Many concurring studies have shown that honey has been tested with E. coli and Staphylococcus aureus to treat other infections, but no studies have shown any evidence of using honey on Staphylococcus epidermidis. This bacteria is typically a skin bacteria, but it also causes infection from implant devices and can be found in catheter-related bloodstream infections. The use of honey can potentially cure these infections caused by Staphylococcus epidermidis. Antibiotic resistance has become a growing problem because many strains of bacteria have become resistant to antibiotics, or in other words, it has become used to the bacteria and the antibiotics will no longer fight off infection. So therefore, there is a growing demand for new ways to treat infections. The side effects of honey are not a common occurrence. In one study, only one patient out of the 50 tested reported a peppery sensation when using honey on their ulcers. Also, there's a small risk of infection in wounds due to honey containing some spores. However, these side effects can be reduced by using a specific gamma-irradiated honey, which is a process that uses radiation to protect the honey from microbial contamination. And this honey can kill the spores while maintaining its healing properties. Despite these few limitations, honey is overall a safe and very effective treatment. It is concluded that honey has less side effects than most over-the-counter ointments and antibiotics because it is natural. After reading the literature, I can pose my own research question of which type of honey will exhibit the greatest inhibition against Staphylococcus epidermidis. My test hypothesis states that if Manuka honey is added to Staphylococcus epidermidis, then it will have the greatest effect of preventing bacterial growth. My null hypothesis states that if different types of honey are added to Staphylococcus epidermidis, then there will be no effect on the bacterial growth. Different types of honey is my independent variable, which is Food Line brand, organic, and Manuka honey. Bacterial growth is my dependent variable. Petri dishes with just the bacteria that receive no treatment is my control. Nutrient agar, Staphylococcus epidermidis, and the environment of the lab remain constant throughout my experiment. To understand how I carried out my experiment, I first gathered 40 petri dishes, cotton swabs, paper discs, and types of honey and bacteria. I then randomized my sample and labeled each Petri dish and then carefully streaked each dish with bacteria. I used the disc diffusion method where I filled beakers with the different types of honey and saturated 10 paper discs in each honey. Using sterilized tweezers, I then placed a disc in the center of each Petri dish. My sample size is 40 with 10 Petri dishes with just the bacteria, 10 food line, 10 organic, and 10 with medical honey. Once my treatments were finished, I then put them in the incubator at 37 degrees Celsius for 48 hours. I then measured the zone of inhibition from each treatment group and put my raw data in an Excel spreadsheet. Then I conducted my statistical analysis. In the left image, my control group shows complete coverage of Staphylococcus epidermidis. In the right image, the food line brand shows little to no zone of inhibition, which means the honey did not inhibit the bacteria. In the left images, the organic honey shows small zones of inhibition along with an unknown substance surrounding the disc. In the right images, Manuka honey shows the greatest zone of inhibition, which means the honey inhibited the bacteria surrounding the disc. For my results, I used a one-way ANOVA test to find the significance of my data. Using the significance level of 0.05, my null hypothesis was rejected with a p-value of p being less than 0.001. To visually display my data, I used a bar graph to represent the results of the different groups of honey. The graph shows the average zone of inhibition in millimeters of each honey, food line, organic, and Manuka. This bar graph shows that Manuka honey has the largest zone of inhibition. There is a clear significance between the Manuka honey group compared to the other types of honey. At the 0.05 significance level, there is sufficient evidence to conclude that Manuka honey is the largest inhibitor. Since Manuka honey contains methylglyoxal, which is unique to Manuka honey only, it provides higher amounts of glucose and fructose, which differs its sugar concentration from the other types of honey. This makes Manuka honey inhibit the most bacteria according to 3 of my studies. Alam et al. Discussed how Manuka honey can be used as an alternative to antibiotics. Since Manuka honey has shown significance to kill bacteria, it could possibly be used to fight off bacterial infections. Since there is an increasing resistance of antibiotics to bacteria, Manuka honey can potentially be used to combat such infections without growing resistance to the honey. The natural ingredients of honey make it safe to use rather than unpredictable side effects of most antibiotics. Honey is also inexpensive and accessible to everyone. It is the priciest type of honey, but still cheaper than most antibiotics. Pertaining specifically to my study, infections caused by Staphylococcus epidermidis, such as surgical wound infections and infections from foreign devices in the body, could potentially be treated with Manuka honey. This is inferred since my study showed a statistical significance of the effectiveness of Manuka honey when used as an antimicrobial as compared to the other types of honey. One limitation present in my study was the unknown substance that grew on the disc infused with organic honey. This could have potentially made an error in my results since I measured the zone of inhibition starting from the substance instead of the center of the disc. If I had access to more tools, then I would be able to uncover what the substance was and how to prevent it in future research. Without these tools, I could use a different brand of organic honey and see if the results come out differently. With further research of antibiotic resistance, researchers could be able to expand treatment options and find ways to make healthcare affordable to everyone. My experiment focused just on bacteria, but if research were conducted studying the effects of honey on patients with staph infections instead of petri dishes, then research could examine the in vivo effects of honey, or in other words, the effects of honey on a living organism. The future of medicine can possibly use honey by itself as a topical or mix it with other compounds to heal wounds and kill bacteria caused by infections. My research has confirmed my career path as I am interested in studying biology and would like to help others find natural alternatives when it comes to the medical field. The research process has helped me develop grit and has made me have better time management skills as it has been a lengthy and rigorous process. I want to give a huge thank you to Ms. Long for guiding me through the research process. I want to thank Ms. Dillon and again Ms. Long for preparing the petri dishes with agar and assisting me in the lab. I also want to thank Dr. Bocock and Dr. Jones for approving my research project. At this time, I will take any questions. Yes, Mr. Mayor.
So I noticed that 4 or 5 of you all have used products in your research. Did you have a budget that you worked with, or was that considered?
Mr. Hare asked if we had a budget to work with, and for the bacteria that we ordered, we did have a budget for the bacteria, but like the honey that we used was our own budget and what we were willing to spend of our own money. But the bacteria was covered by P1 Governor School.
So what helped you decide to use Manuka honey and not another type of honey?
So Dr. Jones is asking what made me decide to use Manuka honey, and Manuka honey has been on the rise in recent studies and has been well known to heal these wounds, and Manuka honey just has been more researched recently, so I decided to use it and see. It is pricey, so I was going to see if it could work, like if you could use a cheaper option, like the Food Line brand or something else. Mr. Shaw.
Can you reiterate what the difference between each type of honey was in terms of their composition?
So Mr. Shaw is asking what the difference between the honeys that I used were, like their composition. And Manuka honey it contains methylglyoxal, which makes it have a more sugary substance. And it's also from New Zealand, so it's the plant that it comes from that the bees pollinate or whatever. And the organic honey, it is, so the plants that they use don't have any pesticides, and the bees that pollinate the plants don't have pesticides on the plants. So that's what makes it organic. And then the Food Line brand, I'm not positive what was in the Food Line brand, but it was probably synthetic or processed. Yes.
So I have a follow-up question to that, because I was curious about the nature of the Food Line honey. Was it in any way processed, or the healthful benefits of it reduced in some manner by being irradiated or heat treated or pasteurized or any of those things? Do you know?
So Miss Waltz is asking if the Food Line brand honey is processed in any way, and I'm not so sure, but reading the ingredients label, you can definitely tell that it's cheap and for like tasting purposes. Yes.
I just have a curiosity question. In Liberty, did you find that this was used for topical only, or the Manuka honey could be ingested for antibiotic treatment?
So Ms. Probst is asking if I found if it could be ingested or if it's just used for topicals. And none of my studies mentioned ingesting it, but because it just focused on the healing of wounds. However, based on my knowledge, it has been on the rise to be using it, mixing it with other stuff to potentially be used to inject it, like in a pill form, but mixed with other stuff, not just honey.